Meow. Meow.
Welcome to our lab. We're working on a fascinating project to enhance egg size in chickens by utilizing ostrich genetics. Let me walk you through the process step by step. So, step 1. Isolation of genetic material. So, the first step is isolating the genetic material. Our objective here is to extract the specific gene responsible for a larger egg size from the ostrich, like the phytogenin gene, which plays a key role in egg yolk formation. We carefully isolate the ostrich DNA to ensure we have the material needed to identify the target gene. Step 2. Restriction enzyme digestion. Next, we use restriction enzymes to cut the ostrich DNA at specific locations. These enzymes act like molecular scissors, allowing us to isolate the gene responsible for egg size. These steps ensure that we have the precise gene segment needed for the next phase. Step 3. PCR amplification. The third step is PCR amplification. PCR, or polymerase chain reaction, is a powerful tool that enables us to make millions of copies of the target gene. By amplifying the ostrich gene, we ensure that we have enough material for the final step, inserting it into the chicken DNA. These three steps, DNA isolation, restriction enzyme digestion, and PCR amplification, are critical to our goal of combining ostrich and chicken genetics. By completing this process, we take one step closer to creating chickens capable of producing larger eggs. Let's move on to the next step, ligation, which involves attaching the target gene to a vector. The amplified gene is then inserted into a plasmid or another vector, which will transfer the gene into the chicken cells using a ligase enzyme. After that, we will proceed to the insertion process using a host cell, utilizing techniques such as electroporation or microinjection to introduce the new gene into fertilized chicken cells or embryos. At this point, we are getting closer to the final product. And for our last and final step, we can now choose the altered cell or embryos that now possesses the ostrich gene, and keep an eye out for a successful gene expression known as the isolation of recombinant cell. This allows us to determine which chicken have successfully integrated the gene. Science is really fascinating, isn't it? Feel free to explore with the other organisms as well, as long as you do it safely and supervised. Thanks for tuning in. Till next time, folks.